pizza's gone, so that means we get started. So everyone, welcome to the Cancer Center Symposium. It's my great pleasure to introduce Eric Pietras as the speaker. So Eric did his graduate work at UCLA. I should mention after undergraduate work in Illinois. And he worked on basically inflammation. I think it's pretty fair to say that Eric has had an interest in inflammation since kindergarten, because he really seems to have this obsessive, interest in it. So he basically worked on how mid-88 signaling links TLRs, the toll-like receptors, and basically bacterial-type pathogens to signaling and the host response. Then he went on and did a postdoctoral fellow with Emmanuel Passagay at UCSF and further studied the roles of inflammation, but in this case in terms of how they affect hematopoiesis, particularly with a focus on hematopoietic stem cells. And he really showed very nicely that hematopoietic stem cells are incredibly responsive to signals such as through the interleukin-1 and other cytokines, and that this can have pretty massive effects on lineage commitment, but also in the ability of these HSCs to self-renew and be maintained. And so now, starting just over 2 years ago, he came to our campus and joined the Division of Hematology as an assistant professor. And he's been continuing these studies, but now he's really moving more into some of the other aspects of inflammatory signal, how it influences autoimmunity, how it influences leukemia predisposition. And so, and with that, he's actually been collaborating a lot with my lab. So it's a great pleasure to introduce Eric, and welcome. Eric, are you going to use this or the...
Yeah, I have to use this.
Okay.
Yeah, I can't do my usual shtick where I put the lavalier and wander around because the lavalier wasn't charged. So please, everybody, always put the lavalier back on the charge stand. Anyway, so it's a pleasure to be able to talk about a really interesting story. We're in debt to a number of individuals, James certainly not the least of whom, for both laying the groundwork and also working as an outstanding collaborator on a project that I think will give us some very important insights into what is emerging as a very interesting syndrome called clonal hematopoiesis of indeterminate potential. So the story I'll tell you is something that we just submitted an R01 on, so it's fresh in my mind, to say the least. What we'll do is start off with a little historical perspective. So this is Rudolf Virchow. He is considered by many to be the founder of modern pathology. So how does that play into a talk in the year 2018 about hematopoiesis and clonal hematopoiesis? Simple. In 1847, he characterized an overproduction of white blood cells as being what we now know to be leukemia, along with another investigator in the UK. He may be the precursor of the idea of the leukemic or cancer stem cell in thinking that cancers arise from activated cells. It wasn't clear what an activated cell was at the time. It was barely clear what a cell was, but nonetheless, the idea that dormant cells could become active and be a reservoir for a cancer. He also proposed in 1863 what's called the chronic irritation theory, and this is more than just meaning somebody is in your office talking too long at you or something like that. What it actually refers to is the idea that a carcinoma or a cancer was associated with what looked like inflammation, a chronic inflammation that recruited macrophages and other cell types that an early pathologist could pick out as being an inflammatory cell. And so the idea was that there was this infiltrate at sites of cancer, primarily solid tumors, that reflected a chronic inflammation. I'm known for giving color commentary in my journal clubs about people who we talk about, so why not here as well? So Virchow was an ardent anti-Darwinist, not for religious reasons. He just couldn't wrap his head around the idea that there's such thing as an intermediate between an ape and a man. He is also the only known human to use a sausage as a dueling weapon. This is not just because he's German, but rather the idea was he told somebody who was offended by his comments about Emperor Bismarck that we'll have a duel with sausages. One of them will be infected with trichinella, which is surprising because he also didn't really believe in the germ theory of disease. But the main takeaway point with Virchow is he really set a framework for understanding cancer and oncogenesis, really not just from a standpoint of an oncogene simply turning something on that leads to a self-reinforcing loop that ignores its environment. Rather, he was making an argument that the environment was actually a critical part in the development of malignancy. So Clonal hematopoiesis, I should say right away, is not a malignancy. Rather, what it is it's a phenotype, or some people call it a syndrome, which is characterized by the clonal expansion of hematopoietic stem or progenitor cells, it's not 100% clear which, that carry mutations typically associated with myelodysplasia, acute myelogenous leukemia, or myeloproliferative neoplasia. So basically these mutations here, TET2, DNMT3A, ASXL1, JAK2, etc. So you have essentially what begins as a heterozygous mutation in an individual stem cell or progenitor that over time expands in the bone marrow and shows itself in the peripheral blood as an allele frequency typically around 4% or above. Now, what does this actually mean for an individual? Well, it does not necessarily indicate that somebody will come down with a myeloid leukemia. Indeed, the progression of individuals who have CHIP to myeloid leukemia on a per year basis is fairly low, and myeloid leukemia still is a relatively rare condition compared to some of the things I'll discuss. So who actually has clonal hematopoiesis? It seems to be associated with specific groups that are kind of indicative of poor health or physiological decline in some way. So that includes older individuals, usually 60 or above, where you start to see CHIP emerge. Individuals who have had cancer, so they've had prior chemotherapy or radiotherapy, it's also associated in those individuals or with individuals who smoke. An important point here, though, and this I think plays into the rest of the talk, is that these mutations are not exclusive to individuals with CHIP. Indeed, sequencing studies that go very, very deep into allele frequencies of like 0.0003% have shown that you can actually find rare clones of hematopoietic cells that carry exactly the same mutations in over 95% of samples that are considered healthy individuals. I think in the particular study it was nurses who we imagine are healthy people. So what does it mean, though, for you physiologically? Because again, most of us have this mutation, but only a few of us will at some point actually see it emerge as CHIP. Well, the first studies to characterize it beyond simply characterizing the presence of these mutations show that it leads to an increase in all-cause mortality. Some of this is related to myeloid hematological malignancies, so there is increased risk for that. But what I think has become most striking in the last couple of years is an appreciation that beyond just malignancy, CHIP is actually setting people up for one of the most prominent causes of death and disease in the United States and around the world, which is heart disease. So 1 in 3 annual deaths in the U.S. are due to heart disease. That's about 800,000 people, according to the most recent statistics in circulation. 790,000 have heart attacks annually. If you look at what clonal hematopoiesis means for your heart, those data are shown down here. And it's a little bit tough to read, but what you can see is that your odds of coronary heart disease increase by at least twofold. Your odds of early onset myocardial infarction, in other words, heart attack, increases by fourfold. In fact, if you look at the numbers with mutations for that, out of 43 total people carrying a CHIP mutation, 37 had an early onset myocardial infarction in one cohort. It really strongly suggests that this abnormal hematopoietic phenotype can lead to some significant downstream morbidities that maybe aren't related just to leukemia, but instead are related to some other deregulations that occur commonly in aging and things like that. In fact, it's actually enough to have made the New York Times at this point. This individual here, Mr. Geer is his name. It says, before being diagnosed with CHIP, which, by the way, it's not really people going to get diagnosed for this, Mr. Geer had run 8 marathons. Determined not to let the diagnosis overshadow his life, he hopes to complete a triathlon soon. I somehow suspect Mr. Geer is not in the highest risk group for actually getting a heart attack, given he's a triathlete. At the same time, what we're starting to understand is that CHIP leads to certain insidious phenotypes that can actually predispose perhaps even healthy people to be more vulnerable for coronary heart disease. So how does CHIP actually emerge? And this kind of gets into a larger question of how does somebody go from a single cell carrying a mutation in any sort of cancer, but certainly for us in the blood, to actually having a leukemia or having a proper cancer? The general idea has been that when you have an oncogenic mutation, what it does is it creates a fitness advantage independent of everything else. That's kind of dogma. And so what happens is you get expansion of that particular clone. This is a good example. So this is a paper that's almost a who's who of the leukemia biology field. And what they did is they took mice that lack TET2. So normally what happens in CHIP is you have a loss of function mutation in at least one allele of TET2. In this case, they model it with mice. They irradiate the mice, and that'll become important later. And then they transplant in, by competition, some bone marrow from a normal mouse versus bone marrow from a TET2 deficient mouse. And whether it's heterozygote or homozygote loss of function, it doesn't matter. What they see is the TET2 deficient cells always win. And so their interpretation is said here. We found that TET2 cells had enhanced repopulating activity in competitive reconstitution assays consistent with enhanced HSC function in vivo. So the idea that these cells carry a TET2 mutation and now are automatically endowed with increased fitness that allows them to expand over time. But we have to go back to the question of, okay, if that's so, then why do 95% of healthy individuals carry a TET2 or DNMT3A or ASXL1 mutation in a vanishingly small clone of their hematopoietic system, and they don't see it expand until they either become old or until they become irradiated or treated with chemotherapy or have any number of other potentially adverse effects happen? So this makes us think a little bit more about something which is familiar to many in the lab, particularly James D. Gregori's lab, because this really does owe a debt to him. What I've put up on top is the frequency of individuals getting clonal hematopoiesis. Down here is a graphic from one of James's papers, which describes how it's called basically a theory of adaptive oncogenesis. It describes that changes not just in the clonal landscape or in the mutation, but rather changes in the environment are what propagate oncogenesis. So in this sense, most of us have all the mutations we're ever going to have, usually by age 30, but most of us don't get cancer, and certainly most of us don't get CHIP if we take this diagram up at the top as evidence. Instead, what happens when you get older, for instance, is you undergo physiological changes which are now no longer selected for. You've already had kids and things like that, and so now you just start to age. So what happens then is you get decreased bone marrow function. You have a process called inflammaging, where essentially senescent cells begin to pump out inflammatory cytokines all throughout your tissues, and that's in large part related to tissue senescence. And so what ends up happening is as your tissues decline, you end up now creating an environment that can positively select for clones that are potentially resistant to these inflammatory effects. And indeed, if we think about what it means to be aged or what it means to be exposed to a genotoxin or something like that, a lot of this falls into the realm of cellular senescence. So cellular senescence is normally a protective response. And indeed, the fact that we can grow to be 30 years of age and have a large number of mutations and not have cancer is probably in large part owing to cellular senescence. So what happens is when you have DNA damage, you engage cell cycle checkpoints like P53, you start turning on cell cycle inhibitors like P16, RB, and P21, and you also start making pro-inflammatory cytokines. Now, this has evolved as a tissue repair response. So you start making what's called the senescence-associated, I believe it's a serum phenotype or secretory phenotype. And among the cytokines that are necessary to initiate this phenotype is IL-1. Both alpha and beta subtypes have been implicated in this work. In addition, you have a number of other downstream cytokines which are produced that, again, can recruit immune cells, particularly innate immune cells, to a site of damage and affect repair. However, at the same time, once you get to this stage of constant physiological decline or constant DNA damage, what you end up having is an unresolved kind of senescence where larger numbers of cells are senescent. They're producing SASP cytokines. And so essentially what you're doing is you're altering the microenvironment to be essentially like my front and back yards back at my house. So the front yard, I replanted it last year. I'm extremely proud of it because it was total dirt when we first got our house. But now it looks a little bit like this. And meanwhile, our backyard, it is exposed to too much sun. It's kind of inflamed in a literal way. And it looks a little bit like this. So where do the weeds grow? The answer is they grow primarily in the backyard. And so we can think of bone marrow by analogy in the same way, in the sense that a mutant HSC, if we think of that as a dandelion, has essentially the same intrinsic fitness no matter where you put it. But you tend to see fewer dandelions in the healthy yard, and the reason is because you have other healthy elements there that can essentially crowd it out for resources, space, etc. Meanwhile, in the nasty yard over here, which maybe is analogous to clonal hematopoiesis bone marrow, what you have is the ability for these cells to now grow independently of some of those resources. So in essence, the intrinsic fitness of the weed doesn't change, but the environment does. And I think that's the central thesis that we'll try to run through this work. So what does that mean in terms of aging and irradiation exposure? Well, in both cases in the hematopoietic system, you see a sharp decline in the fitness of the hematopoietic stem and progenitor cell compartments. So this is work from James's lab where mice were exposed to irradiation, and then 9 weeks later they were transplanted. So basically a 9-to-1 ratio of competitor to irradiated marrow. And what you find is the irradiated marrow loses. On the other side of things, in the aging context here, what you see is young versus old HSCs, purified HSCs in this case, transplanted into recipient mice. And what you find is that on a per HSC basis, the old HSCs have greater trouble reconstituting the animal. They have decreased fitness. And so what this leads us to is a model where we can imagine that inflammation is largely a good thing. We need it in order to fight off pathogens and things like that. But when you put it in the context of a senescent phenotype or a tissue decline that you might find in aging or exposure to chemotherapeutics, what you end up getting is basically an expansion of the lineages of cells emerging from here, such that now you get clonal hematopoiesis. So that's our model to work from. How do we know that this might actually be the case? Well, what we've done is we've tried to model a number of different scenarios in which clonal hematopoiesis is prevalent. So what you see here are basically graphs showing IL-1 alpha production in the bone marrow of mice that are exposed to different insults. So these mice were simply aged for 24 months. You see a significant increase in IL-1 alpha. Next to that, you have mice that were exposed to irradiation, and then 8 weeks later their marrow was interrogated. You have more IL-1. Next, you have mice that were exposed to 5-FU and looked at 2 weeks later. Again, more IL-1. And then lastly, even when we model the presence of IL-1 just by injecting IL-1, what we find is that gets translated into additional IL-1 production in the marrow. So we're giving mice IL-1 intraperitoneally, and what we find in the marrow is IL-1 alpha being produced, presumably by some of the stroma, but maybe by some of the other immune cells. And so for us, IL-1 has been significant for a number of reasons. One is it is long associated with hematopoietic changes that both mediate repair and that also can mediate disease pathogenesis. So IL-1 itself is a pluripotent pro-inflammatory cytokine. It was characterized by Charles Dinarello, who is faculty here, as originally called human leukocytic pyrogen. There are two subtypes of IL-1, alpha and beta. The differences lie in how they are secreted as well as what cell types express them. So those regulations will dictate some tissue specificity. They both bind to the IL-1 receptor and both mediate largely the same activity. They are produced upon nearly every injury or insult or infection that you can imagine. IL-1 is a common denominator of nearly every inflammatory process, and that includes senescence, where again, based on work from a few different labs, what happens is you have IL-1 production in order to elicit the rest of the SASP. Judy Campisi's lab has particularly pioneered that work. And lastly, again, it's a driver of many of the chronic inflammatory diseases that are treated in the clinic, in many cases with IL-1 blockade, and there's a large number of them. The other interesting point is that clonal hematopoiesis mutations themselves can also create their own IL-1 environment. So in particular, TET2 deficiency makes macrophages extremely sensitive to oxidized LDL. So you wonder why there's a connection with heart disease. This may well be it. atherosclerosis is now considered a chronic inflammatory disease. And when you have macrophages that are knocked out for TET2, they produce much more IL-1 beta in response to oxidized LDL and other inflammatory cytokines, suggesting that TET2 mutation itself could also create a feed-forward loop where you continue producing more IL-1 and continue to derange the bone marrow. And then lastly, and I think this is an important point, IL-1 blockade is a therapy that has existed for well over a decade now. It's something that's clinically tractable, and it's something that has been shown both to reduce cancer and heart attack risks. A recent study by the CANTOS trial group showing precisely this, that you can even get decrease in lung cancer in individuals who had an initial heart attack and were put on an IL-1 beta blocking antibody. This basically reduced the risk of lung cancer in addition to other sorts of comorbidities. So the questions that I'll address today during the talk are two. One is, does chronic IL-1 impair normal HSC fitness? So the idea being here, if IL-1 is a key component of the SASP that's produced in chip-associated conditions, what is its impact on normal hematopoiesis? Does it take the healthy lawn and basically make it the bad lawn? And then secondly, do HSCs that actually carry chip mutations respond differently to this pro-inflammatory context? So do they function as the weed whose intrinsic fitness doesn't change regardless of the environment? For this, my lab specializes in understanding hematopoiesis, and so what this means is we try to understand how the hematopoietic tree itself is regulated and how HSCs can be essentially altered in order to generate different lineage distributions. So this is the classical model of hematopoiesis where at the root of the tree, assuming that the root is facing upwards, you have the hematopoietic stem cell. This cell self-renews, so it can make a copy of itself when it divides. And it's also quiescent, so what that means is long-term, HSCs are basically taken from the they're taken from the cell cycle and they're retained in this quiescent state. It helps limit DNA damage and other stresses that they might encounter because essentially you have a small pool of these cells. They need to last you a lifetime. They need to be protected. So quiescence is the key mechanism by which HSCs are protected from cellular stresses and protected from exhaustion. When they differentiate, they go through a progressive series of amplifying steps because essentially you're going from somewhere between 20 to 100,000 of these to up to like 10 to the 12 or 10 to the 13 red blood cells and platelets. So you need a lot of amplification to move down these lineage trees. And then lastly, the HSC gives rise ultimately to all of the mature effector cells, both in the lymphoid, myeloid, megakaryocytic, and erythroid lineages. In our lab, we use the mouse as a model for this because we have excellent genetic tools and also the ability to enrich for HSCs at high purity. You'll hear me throughout the talk refer to something called a SLAM HSC. What that means is simply that we take cells that have not yet expressed a lineage marker and we subdivide them by some additional markers, c-KIT and SCA1. You'll hear about LSK. So what that is it's enriched for HSCs, but not as much as we then further enrich for. And then finally, we get down to what is an HSC here. In this case, functionally speaking, about 1 out of every 2 of those can repopulate in a radiated recipient mouse. You'll also see that since then, we've also gone further down into that specific compartment and found a long-term HSC, which has been characterized by others and that we use now as an even more rigorous definition. So as I mentioned, HSCs are very powerful cells. They can produce a large number of progeny, and for that reason, their function has to be very carefully controlled. So what does that mean? That means that they exist in a bone marrow niche, which is essentially an emergent property of many different cell types that surround an HSC, including arterioles, endothelial cells, mesenchymal stem cells, even adipocytes and nerve cells as well. And what these do is they basically help guide HSC decisions. So HSCs can decide whether to proliferate and through that process either self-renew or generate a differentiated daughter cell that will eventually become a mature cell. They can leave the bone marrow as necessary, or they can undergo death, or they can also undergo senescence as another phenotype. And so essentially what you have is a collaboration between things like quiescence-inducing factors produced by the niche, as well as survival factors, et cetera. And what this allows is for an HSC to be a quiescent cell with a low metabolic rate. It can efflux toxins, and it can basically keep itself out of cell death unless absolutely necessary. The other key point is that HSCs are not simply cells that exist in a vacuum completely protected from the outside world. They're actually fully responsive to inflammatory cues. And this was the center of my postdoctoral training, and it forms part of the central rubric of our lab now. So this is a paper from 1989 showing that if you irradiate mice and treat them with IL-1, you see a faster recovery of myeloid cells. And this is a key theme. HSCs generate myeloid cells essentially as a first response type of cell, and IL-1 specifically trains them to do so. This was largely understood as a functional output. Then later, during my postdoctoral work, we spent a little bit more time understanding the mechanism. So what happens when you put IL-1 on an HSC is it engages in a myeloid commitment gene program. And so what that means is basically it starts to turn on, there we go. It starts to turn on genes like PU.1, which is a myeloid transcription factor that helps it to rapidly become a myeloid-committed cell. So here what you're seeing is basically Fc gamma receptor and the integrin MAC1. These are considered myeloid commitment factors, and you see that HSCs acquire them much more rapidly in culture when you have IL-1 present. The other tool that we have and that I'll refer to in the lab is we have mice that are knocked in with a PU1 fusion gene. So this allows us to track PU1 activation within HSCs. And what you notice is that when you put IL-1 on HSCs in culture, you basically see them turn on PU.1 and rapidly differentiate. And so essentially what happens is this. You have activation of PU.1, rapid myeloid differentiation, and what that leads to is, especially in cases of acute inflammation, is faster recovery. It's an adaptive response to an emergency need. But what happens in the chronic setting is actually a completely different scenario. Because instead of being an adaptive response to acute need, you now have what seems like a remodeling of the hematopoietic system in a way that might actually be maladaptive. So in this case, we model this by treating mice for about 20 days with IL-1. We give it IP, and then we analyze the bone marrow. These are data that we generated in our lab to confirm our previous results. What we see is an expansion of granulocytes. So these are myeloid cells like neutrophils. We see a decrease in B cells. So you're seeing an accentuation of one lineage, the one that's needed for emergencies, over the lineage that takes a longer time to develop and so can be sacrificed when you have acute need. In addition, when we look at the early immature hematopoietic compartments, what we find is an expansion of these populations that I won't spend a lot of time on, other than to say that they help generate myeloid cells. They seem to function as an emergency pathway during chronic and acute hematopoietic need. On the other hand, the common lymphoid progenitor decreases substantially. And then lastly, just to make the point that we can also trace PU1 activation in vivo using our mice, these are HSCs from mice that were treated up from 1 to 20 days with IL-1. And what you notice is that PU1 is turned on rapidly, and it stays on under chronic inflammatory conditions. So essentially, we have a model where hematopoiesis is being redirected from the from the lymphoid lineage and erythroid lineage into the myeloid lineage, primarily through a fate choice that accentuates these differentiation pathways over this differentiation pathway. So what does that mean for HSC functionality besides an aberrant blood system? In our field, we do this by bone marrow transplantation. It's considered the gold standard for identifying changes in HSC self-renewal properties. So in this case, we purified hematopoietic stem cell containing fractions, and we transplanted them into irradiated mice. And what you notice is that these myeloid primed HSCs that we harvest from the mice differentiate away, and what we find is lower donor chimerism, as you see here in the blue. So those are the progeny of HSCs that were harvested from IL-1 treated mice. In addition, if we overexpress PU.1 in HSCs, and so these HSCs are essentially proliferating HSCs that have been taken out of the marrow, transduced with PU.1, and then put back into a recipient animal following irradiation, again, you see that there is a failure of these cells to generate long-term progeny. So the way we interpret that is that when these cells have become activated, PU1 basically tells them to become myeloid and to do so quickly. And so what happens is they essentially exhaust. And so the overall model that we work with in the lab is basically this one. To simplify, the major point is simply that inflammation can act directly on HSCs, It can turn on specific subsets of HSCs that I won't go into today, but that we feel become very important for balancing blood production. And it leads to overproduction of myeloid cells. Again, that's good in the acute sense. It's probably bad in the chronic sense. So since starting the lab, what we wanted to do is expand upon this and find out a little bit more beyond just transplanting HSCs, what actually happens to the blood system and what actually happens to HSC function in vivo during chronic inflammation. People who have chronic inflammatory diseases do not die of bone marrow failure. It means there's some break to prevent that instance from occurring in a kind of universal level. So we wanted to understand a little bit more of the biology of HSCs in these chronic inflammatory circumstances. So we went back to our mouse model, and one of the first experiments that we did was to understand the cell cycle behavior of these HSCs. This is looking at a couple representative plots. We used DAPI to define DNA content, and then Ki67 to discriminate quiescent cells in G0 versus cells that have become activated and moved into G1. What you notice is that under acute IL-1 treatment, so that's just one injection of IL-1, you actually get a profound amount of proliferation within the HSC compartment. We think this could be a bystander effect related to loss of contact with the niche, but it's still here after millions of years, so perhaps it's also adaptive as a way to supply cells to the hematopoietic system under emergency conditions. However, when we look under our chronic conditions, what we find is that the HSC compartment is back into a largely quiescent state. So continued exposure to IL-1 does not equal continued proliferation, and it makes a lot of sense. If it did, then you would essentially undergo bone marrow failure. You'd be losing a self-renewal signal, and instead you'd be seeing IL-1, which forces cells to differentiate, as we know from our culture experiments. In addition, we find that reactive oxygen species levels in the hematopoietic stem cell compartment are retained at low levels. Again, HSCs need low ROS levels in order to maintain their potency. We find that after 20 days, these cells have the same overall level of reactive oxygen species that they did at day 0. So there's something that's maintaining these cells in this kind of low reactive oxygen species quiescent state. When we look at changes in HSC activation pathways by phosphoflow, what we find is that there's a small decrease in the activation of these pathways, suggesting that something is impinging on these pathways that normally would be turned on by IL-1. And in collaboration with Angelo D'Alessandro and Travis Nemkov, we've also looked at HSC-enriched fractions for metabolites. And what we find is that between a control condition and 20 days of IL-1, there's really no change in overall metabolic state. So these cells, despite chronic IL-1 exposure, are still metabolically quiescent. Looking at the molecular mechanism through RNA-seq, what we noticed is that during chronic IL-1, HSCs downregulate key mechanisms that are related to protein translation, ribosomal RNA processing, tRNA metabolism, and ribosome biogenesis. They also downregulate pathways that are, again, related to cell growth, like MYC, mTOR, AKT, et cetera. We verify these data using Fluidigm, which allows us to sort small numbers of HSCs, because, again, per mouse, we're dealing with 2,000 to 5,000 cells that we can use in this fraction. And we run essentially what's a high-throughput qPCR. And what we find is that key cell cycle regulators like D-cyclins that are necessary to move HSCs from G0 to G1, or key growth pathways like MYC are repressed. Meanwhile, cell cycle inhibitors like P21, which is CDKN1A, are upregulated in this context. So essentially what we're seeing is the hematopoietic stem cell compartment turning on the brakes and preventing itself from differentiating away by downregulating the key genes necessary to actually sort of promote cell cycle entry, and upregulating genes necessary to keep cells out of the cell cycle. We've done further analysis into the enriched long-term HSC fraction via Fluidigm. This is a heat map looking at all 96 genes that we see, and again, we're specifically looking in about 25% of that previous fraction that we've considered long-term HSC. And based on these results, what we've been able to do is essentially validate what we found in the SLAM HSC compartment. Using principal component analysis, we find that principal component 2 here is largely what separates naive HSCs versus long-term HSCs that have been exposed to IL-1. And again, the gene signature here is largely related to downregulation of certain cell cycle genes, upregulation of cell cycle inhibitors, and some other changes in pathways that relate to normal HSC identity. And once again, looking at some of our key genes, we see that the long-term HSC is exactly the reflection of the more general SLAM HSC gate. The other point to mention is when we look in our long-term HSC, we again see activation of PU.1. So by SLAM HSC RNA-seq, we can pull out the signature. In addition, when we look at our long-term HSC fraction and look at PU1 levels, we see that chronic IL-1 turns PU1 on. So we can detect more of it as the EYFP fusion, and we can detect its target, the integrin MAC1. So why is this significant? Well, it took us a little bit of time to try to sort of integrate the gene signature we were seeing with the ability of PU1 to be turned on and activated in hematopoietic stem cells. So PU1 is kind of an interesting double-edged sword, because on the one hand, yes, it is a myeloid differentiation factor. But on the other hand, work from Dan Tenon's lab and others have shown that PU1 can also act as a cell cycle inhibitor. Why is this important? It's important because in order for a myeloid cell to differentiate, you have to accumulate enough PU1 to reinforce the gene program. How do you do that? You slow the cell cycle a little bit so that PU1 can accumulate, so then you can keep moving along the myeloid differentiation path. And so in a proliferating cell, that's going to give you a myeloid gene program. But what happens in a quiescent HSC? What you're doing is you're simply turning on cell cycle inhibitors and downregulating pro-cell cycle gene programs, which will end up resulting in probably an enforced dormancy. And in fact, work that was published in 2016 when I was just finishing up my postdoc, now it starts to become understandable. So we had done some single cell tracking analysis where basically we did time-lapse microscopy on individual HSCs, and we could separate out PU1 levels. And so what you find is when you treat naive HSCs with IL-1, you get more PU1. But what was always puzzling to us is that the very first division that HSCs underwent, their exit from quiescence, so to speak, when we looked at IL-1 treated cells, the time to that division was slower. And even when we looked at naive HSCs that had higher levels of PU.1, what we found is it took them longer to exit quiescence and divide for the first time. So this was evidence that at the time of the publication of this paper didn't make a whole lot of sense to us, but now I think makes a lot of sense. PU1 functions as a cell cycle inhibitor in this case. So yes, it can drive myeloid gene programs, but it can also enforce dormancy in cells that are already dormant. As with our HSC subsets, we also took the PU1 EYFP mice and we subdivided the HSC compartment into its distinct components based on PU1 levels. So we took PU1 high, intermediate, and low cells. And again, we looked for some of these key genes. And what we found in the PU1 high cells was at steady state, increased levels of CDKN1A, decreased levels of MYC and cyclin D1, exactly as we would predict if PU1 is functioning to inhibit cell cycle activity. When these cells are exposed to IL-1, they turn on that program even higher. So not only do they turn on more PU.1 in this condition, but now they also continue to turn on P21, continue to downregulate cyclin D1, and enhance the activation of that cell cycle arrest program. On the flip side, what we find in the PU1 high fraction is kind of an equivocal level of HSC self-renewal genes, but those self-renewal genes now increase in the presence of IL-1. So this leads us to believe that what PU1 starts to do is enforce dormancy. And for that reason, PU1 high cells retain some of their stemness. So we think, again, this is a protective mechanism. To prove that, what Jen Rabe in our lab did was basically to take mice and treat them with IL-1. And again, these are PU1 EYFP. And then take the PU1 high and low fractions and perform bone marrow transplantation using the purified HSCs. And when we first did this experiment, we didn't really think of PU1 in the sense we do now, so the results were confusing, but now I think they're very, very clear. So what you find is that under steady state conditions, it's actually the PU1 low cells that seem to retain most of the repopulating activity. You do see some in PU1 high, and so we think there are some other gene dosage and effects at normal levels of PU1 that are interesting for us to look at. But I think what struck us most is that when we looked at the IL-1 beta treatment condition, what we found is that the HSC activity now was largely in the PU1 high fraction, these dark blue bars here. And when we looked at HSC chimerism, again, the same situation is it was lower than normal, but at the same time, functional HSCs were residing in this PU1 high fraction where we see the cell cycle arrest gene signature. We could even see that by surface phenotype. Again, this gate here is where we find our long-term HSCs. And under the conditions of IL-1, you find that that population is now enriched in the PU1 high fraction. So to sum up this part, we think PU1 functions really in a context-dependent fashion that can both drive myeloid differentiation and at the same time, in quiescent HSCs, can also enforce dormancy. I think it's a great example of evolutionary parsimony. You're doing multiple things with a single gene rather than making a new gene in order to do a completely different thing. So essentially, an HSC that has become activated will use PU.1 to differentiate. Maybe that explains why chronic IL-1 actually gives you more myeloid cells, is over time, any HSC that does enter the cycle and divide goes down a myeloid lineage. On the other hand, for the rest of the HSC compartment, PU1 is functioning more as a cell cycle inhibitor. It's downregulating proliferation. We're testing right now if it downregulates protein synthesis. And again, we see some of the key genes that are necessary to drive HSC activation turned down in this context. Another point to bring is that when we look at conditions of irradiation or conditions of aging, in this case looking at data through the help of Kelly Higa from one of James's papers, as well as mining some old data from microarrays that I ran in Emmanuel's lab, what we find is that once again, you downregulate in these chronic inflammatory conditions the gene programs necessary to enter the cell cycle. What you upregulate is PU.1. So what we're starting to see now is essentially the basis by which we can understand decreased HSC fitness, because ultimately fitness is a competition between cells that divide more and generate more progeny versus those that don't. So if you're enforcing dormancy in a particular cell type, you have the potential, yes, to save it from exhaustion, but when you have a cell that perhaps ignores that signal or doesn't respond to it correctly, what you're going to have is a decrease in relative fitness. So a couple of key points of what we're working on now in this project is, first of all, to understand if PU1 really is a brake against stem cell exhaustion. So what we're doing is a knockdown experiment in order to understand if we bring it back to normal levels, do HSCs go away? We're also trying to now understand some other aspects related to HSC heterogeneity. So do certain HSCs turn PU1 on more, and what happens to them? So we've got single-cell RNA-seq and other lineage tracing experiments to try to identify how the HSC compartment contends with chronic IL-1. So back then to our model of how we think clonal hematopoiesis may work in this context. So the idea is that somehow the mutation that occurs must be advantageous for a mutant hematopoietic stem cell, and that leads to its ability to expand under these inflammatory conditions. So the work that I'll show you is owed in large part to Kelly Higa in James's lab, just as all the work I showed you before that is owed in large part to Jen Rabe in my lab. And so what I want to show you is basically the model we use. So we have mice that are conditional knockouts for TET2. And what this allows us to do is inducibly, through the treatment of the mice with tamoxifen, remove TET2 alleles from hematopoietic stem and progenitor cells. And then what we can do is we can transplant them. So we can essentially model what would happen in a human who has a rare clone that is TET2 deficient, whereas the rest of the bone marrow is going to be normal. And so we can do this both in heterozygous as well as homozygous knockouts to model the gene dosage effects that we can see in CHIP. And then finally what we do is we treat them with IL-1 and we ask the question, who wins? So the first experiment, again, what we do is we basically compete our TET2 deficient HSCs against what is simply a congenic boy jay. It has a different CD45 marker. It just allows us to track its lineage. But what we do is we transplant them into the same recipient mouse. We use a conditioning regimen that uses busulfan and CD4, CD8 depletion this is a less inflammatory treatment than what you saw when we look at the bone marrow of mice that are treated with the radiation. You don't have that chronic IL-1 response that everybody else is trying to interrogate. Instead, what we can do is now give mice IL-1, and once again, using donor chimerism in the peripheral blood, determine who wins this competition game. And the results are really quite striking. So contrary to what has been published, what we find is that when you don't have IL-1, you really don't see any sort of competitive advantage to having a TET2 mutation, whether it's homozygous or heterozygous. It's only when you actually add the inflammatory stimulus that in all of these irradiation experiments, like the one I showed you earlier, is already there, that you now see the competitive advantage that is endowed by TET2 deficiency. And here now, you see the expansion of the TET2 deficient compartment. So it's a striking result because, again, it says how we as a field use irradiation so commonly to condition our mice that we forget what that irradiation can do in cases when you have a non-normal hematopoietic stem cell. And so I think as a field we see these data, and I think it causes us to rethink a little bit how we interrogate hematopoietic self-renewal. On top of this, Kelly did another experiment, and this is also very ingenious because the question is this due to the fact that IL-1 is acting directly on HSCs, or is it due to the fact that IL-1 is acting indirectly elsewhere? So what Kelly did is model the bone marrow in a dish and basically create a culture experiment where you're competing one-to-one, in this case the LSK HSC-enriched fraction, plus or minus IL-1. And then after 10 days, you're looking at how many of these cells still retain that immature phenotype. Because again, self-renewal means maintaining an immature HSC-like phenotype. And once again, the result is quite striking. So here, once again, you're looking at the LSK compartment, and what you find is that in the absence of IL-1, every genotype is just as good as the other. You add IL-1, however, and now in these proliferating cells, what you're doing is you are forcing the wild-type cells to differentiate, so you're losing your LSK. And on the other hand, the TET2 cells seem largely insensitive to this effect, and they still retain a higher level of LSK. So how can we understand this from a molecular perspective? Because essentially what's happening is it seems as if TET2 deficient cells largely don't care that IL-1 is present. In fact, they seem to be able to use it as a proliferative or expansion driver. So we used our Fluidigm system to interrogate what these different HSCs look like. And here I'm just comparing the TET2 wild types versus TET2 heterozygotes. And so the data you're seeing on the graph is the heterozygote relative to the wild type. And this is interesting because at steady state, there's really nothing here that suggests a TET2 deficient cell would have any advantage over a wild type cell. In fact, TET2 deficient cells already have higher levels of P21, which is a cell cycle inhibitor. That suggests they might even be less adapted to be able to expand in this environment. On the other hand, when we treat with IL-1, in both cases we see Pu.1 turn on. And now what we're looking at is the relative expression level of these genes in, once again, TET2 heterozygotes versus TET2 wild type in the presence of IL-1. And so now, just like in the culture and in the bone marrow transplant experiment, the advantage for TET2 deficient cells becomes apparent. So what we see is that TET2 deficient cells don't downregulate cyclin D1 as much, and they don't turn on cyclin or CDKN1A as much. So these two key Pu1 repressive and activating targets are not activated or repressed to the same level as they are in wild type cells. This would automatically give an advantage to a cell that can proliferate even just a little bit more. At the same time, we look at HSC self-renewal genes, and once again, now we see an advantage in the HSC compartment, which is TET2 deficient. So once again, the winner is the one that can self-renew, generate more copies, and continue to generate more blood. The TET2 heterozygous cells can enter the cell cycle more based on these data, and probably can self-renew more in order to clonally expand in the bone marrow. And so basically what our model is that we're seeing the effect of an epigenetic dysregulation. That would be my hypothesis here. But our model is that in order for a TET2 deficient HSC, and perhaps all CHIP-associated mutant HSCs, in order for them to expand, they need a pro-inflammatory cytokine to tip the balance in their favor. And so what happens is the normal cells undergo a growth arrest program. They're obedient to this signal. They basically turn up cell cycle inhibitors, turn down cell cycle activators, and stay quiescent. It's what they're adapted to do. Meanwhile, when you have an aberrant epigenetic change, because TET2 is a regulator of DNA methylation, now you get still the same program, but a weird caricature of it. You're not getting the same genes turned on to the same levels. And instead what's happening is now you have an emergent clonal advantage for these cells that wasn't there in the absence of IL-1. And this might explain, again, why CHIP seems to be restricted to these specific cohorts of individuals who have these chronic inflammatory or senescent bone marrow phenotypes. And so lastly, the way to think about it basically in terms of just a sentence is it's the honey badger versus the ostrich. This is kind of how we think about it. So the honey badger doesn't care. I didn't realize that meme was actually as old as it was. I thought it was like from 2 days ago. But so the honey badger doesn't care. That's your TET2 deficient cell. It sees IL-1. It turns on something like a program, but it doesn't affect its self-renewal. In fact, it gives it an advantage. Meanwhile, the normal HSCs stick their heads in the sand. They try to retain quiescence and dormancy, and so they don't generate the same amount of output. And so this leads to clonal expansion of the mutant clone. So what we're doing now is we're basically trying to understand whether cell-intrinsic IL-1 receptor signaling is required for this. Again, it's the idea of setting up for being able to use something like an IL-1 blockade therapy in the context of CHIP. And indeed, what we ultimately are doing in collaboration with Charles Dinarello is to treat irradiated mice, where we know we have this chronic IL-1 and senescence, with IL-1 blockade, both anakinra as well as anti-IL-1 antibodies, to understand whether we can either reverse or limit the clonal expansion of these mutant cells. And so lastly, just to conclude and provide some implications for this, so chronic IL-1 activates what we believe is a PU1-dependent growth arrest program in HSCs. Right now it's PU1-associated until we do the knockdown. This mechanism, we think, is necessary and has evolved to prevent HSC exhaustion using a sort of parsimonious evolutionary approach. TET2 deficient HSCs, on the other hand, fail to activate this program correctly, and so that leads to their preferential ability to expand under the influence of chronic inflammation. And then lastly, what does this mean? Well, first of all, it changes our understanding of oncogenesis along the lines that James has really been pushing for a number of years here. This is not a process where simply an oncogene endows a hematopoietic cell with a massive amount of self-renewal capacity that can just overtake a marrow. Instead, it's a process of somatic evolution that is driven by changes in the environment, in this case, chronic inflammation. It suggests that blockade of chronic inflammation can, again, reverse and limit CHIP, and so this will be one of the most exciting things that we can now do in the lab. And then lastly, the key point is tractable therapeutic approaches to block IL-1 already exists. The CANTOS trial using an anti-IL-1 beta antibody is a great example. Years and years and years of anakinra use in a multitude of chronic inflammatory disease suggests that we already know how to use the drugs that might actually be impactful in this case. And once again, if we're thinking of heart disease as being a leading cause of death and CHIP being a major contributor, then I think this becomes a very important therapeutic modality to explore. So lastly, I would like to thank an outstanding group of young investigators that I have the privilege to work with, particularly Jen Rabe in the lab. She's responsible for much of the work that you saw in that first part within a short amount of time. She has broken my record for the sheer number of mice transplanted at once by about 5-fold. So things were easy for me as a postdoc. Jen has really just taken this to the next level and has generated an amazing amount of interesting data. Also, Taylor Mills, who is working on another interesting project related to what HSCs remember from chronic inflammation, and James Chavez, who is a brilliant young technician who is hungry for a scientific career and does outstanding work in the lab. Also to thank Giovanni Hernandez, who set up a lot of the foundation for my lab, and he's now teaching English in Japan. Also, a number of collaborators with whom we cannot do this work without. That includes Craig Jordan, the division head of hematology, James DeGregori, and Kelly Higa, certainly, who have been outstanding collaborators and are both reasons that I came to this university. Angelo D'Alessandro and Travis Nemkov have been our collaborators in the metabolomics space. We continue to generate interesting data with them that I think will figure largely into this work. And also Charles Dinarello and Ike-Jun Tan, who does a lot of our bioinformatics support. And then lastly, collaborators elsewhere for RNA-seq and single-cell tracking. And finally, funding sources from the Betcher Foundation, NIDDK, and the RNA Bioscience Initiative. Happy to take questions. Thank you. Yes?
Very intriguing work, especially when you are talking about was wondering if you would have done it actually one day, would you have gotten the same results that we're seeing is the acute effect that you just mentioned that is already there from the camera? Did you comment on that?
So I will say that is a really interesting question because, we have this interesting bystander effect where HSCs briefly enter the cell cycle in response to that initial burst of IL-1. We're trying to figure out at the moment whether that's an artifact of the experimental design or whether that occurs anytime an HSC, sees a chronic inflammatory stimulus. But I think as you point out in your question, it could actually be a useful tool for simply interrogating whether a TET2 deficient HSC maybe responds simply by generating more TET2 deficient HSCs. So another way to imagine the process is if we all go through a lifetime of acute insults, perhaps there's also a case where an advantage is born via that route. So we've not done the experiment, but we can do the experiment, at least to see if there's maybe a higher proportion of cells in cycle. The other thing that we're developing is a tool that will allow us to lineage trace from the most primitive HSC compartment what cells are produced and in what amount. This would be a particularly interesting tool. So what you do is you turn on like a EYFP in that HSC compartment, and then you see where it expands to over time. So if we did that experiment with an acute IL-1 burst, what would be very interesting to find out is do we just see an expansion of TET2 deficient HSCs, or do we see the expansion into some of the non-HSC compartments? Really nice. Yeah, thanks for the idea. Yes?
Do you see in transplants Q1 inducing higher expression of IL-1 itself? Is IL-1 itself hurt?
Yeah, so that's an interesting question. In our RNA-seq data, we don't see IL-1 turning on in the HSCs, and granted, maybe they just don't have the machinery to do anything useful with it. However, what they do turn up is IL-1 receptor, which is kind of interesting. So either they're seeking more signal because of the fact that they're dormant or something else. What we don't know is what the source of IL-1 is yet in either the irradiation environment or in our IL-1 chronic treatment environment, or in aging, for instance. And I think that'll be an interesting question. Whether PU1 is responsible for that induction, we don't know yet, but things like, for instance, in senescent cells, NF-kappa B and mTOR are what turn on the IL-1 alpha, which is secreted by senescent stromal cells based on Judy Campisi's work. So it suggests that it could just be an NF-kappa B response, and that would fall nicely in line with a chronic inflammatory phenotype, given NF-kappa B is a key target for inflammatory signals.
So maybe later on down the line, the IL-1 receptor is actually once the cell leaves.
Yeah, so by leaving in terms of basically differentiating or something like that, yeah. Yeah, I mean, you could imagine that they're turning up antennae so that when they do become activated, they're a little more sensitized to the signal because they think something is amiss. Not to anthropomorphize too much, but that could be a really interesting idea. We've just done some experiments where we've treated mice with IL-1, taken their HSCs and put them into culture to essentially find out, are those cells more actively primed to basically become myeloid by virtue of that IL-1 signal. And we haven't done it with IL-1 in the culture, but I think that's an interesting condition because I bet they're way more activated to become myeloid cells in that case. Yes, Bob. Yeah, so that's an alternative. So they don't have that much of a phenotype at steady state. It's actually one of our alternative approaches in the R01 because P21 is a PU1 target, and under conditions of hematopoietic stress, which have been modeled by David Skadden's lab using 5FU, what you find is that P21 deficient mice undergo HSC exhaustion. So it could well be one of the main relevant targets of PU.1 in this case that maintain HSCs in a quiescent state. It's worth investigating because the mice are available and all that sort of stuff. And it's an easy one gene to, that people are going to obviously ask about. Yeah, Craig.
Eric, I'm guessing for practical reasons that the experiments that Kelly did were using young or relatively young mice, or stem cells relatively young. And one of the things I think I've learned after engaging with many times is that stem cells get older, they're changing a variety of ways, including their myeloid predisposition. So it makes me wonder whether or not these experiments actually underestimate the selective advantage of knockout. You get older stem cells that have more of a myeloid programming
Yeah, Craig, I think there's probably a lot of rationale for believing that, yes, we are underestimating, and a couple of reasons for that. One is the aged bone marrow niche in and of itself is a terrible environment. It has many fewer supporting cytokines. It has, stem cell dysfunction in all of the stromal compartments normally required for maintaining HSCs. So for that reason, something that can be a little more independent, like a TET2 mutation, might, again, have a much stronger advantage. On top of the fact that with aging, you're collecting a number of other factors independent of just IL-1 exposure, stuff like replicative stress phenotypes and cell polarity problems and things like that. So I think that is a good point. Yeah, from a practical standpoint, it's tougher to model, but there's every reason to think that under those conditions, maybe we see even worse clonal expansion. Okay, well, thank you.